Neurogenetics is a relatively new field within neuroscience that focuses on the role of genetics in the human nervous system. As the name implies, neurogenetics applies the techniques of molecular biology and genetics to study the nervous system on a micro level. Researchers are interested in neurogenetic disorders such as Huntington's disease, autosomal dominant cerebellar ataxias, and spinal bulbar muscular atrophy. The goal is to develop theories regarding the pathology and molecular basis for these complex disorders so that viable treatments can be created to stop and/or reduce the likelihood of incidence in the population. Here's a diagram of a human cell. The major organelles were labeled and swim in a pool of amino acids called the cytoplasm. The body synthesizes 11 of the 20 amino acids we need, and those amino acids are sent to the ribosomes, which are massive molecular machines that organize amino acids into peptide chains as instructed by messenger RNA. I should also mention that ribosomes are just larger, more complex proteins, so proteins basically make themselves, like robots eventually will before they take over as the de facto variety of superiority and stuff.
Somehow, I told you so, just doesn't quite say it. Get off my car!
When a cell divides, each of its DNA sequences splits from a double helix into two single strands with the help of helicase enzymes. The Watson-Crick base pairing principle determines how the nucleotide monomers will bind to incoming deoxynucleotide triphosphates. Guanine will bind to cytosine and adenine will bind to thymine, and sometimes uracil. So all cells contain the same set of genetic instructions, but a cell will only produce specific proteins depending on what type of cell it is, which seems oddly convenient. I mean, how do cells know which proteins to create? And the answer is the promoter sequence. Here's a very brief description of how it works. So RNA polymerase scans the DNA for promoter sequences along the 3' region of a gene, which are regions in the outer region of DNA that bind well to the RNA polymerase. Operator sequences are neighboring sequences to the promoter sequences, but are instead bound by transcription factor proteins. Each transcription factor will bind a specific promoter sequence so that the cell can easily decide which genes to transcribe. Approximately 25 to 35 base pairs upstream of the transcription starting site is a TATA box, and when transcription factors bind to the TATA box, the RNA transcription complexes form, which promotes the initiation of transcription. Here's an intuitive way of thinking about it. The cell nucleus contains chromosomes, which are made up of gene sequences, which contain promoter sequences, and as a result, each cell type has its own unique characteristics or phenotype, which are largely determined by the proteins the cell will transcribe. For instance, both glutamate and GABA neurons will produce glutaminase, but only GABA neurons express the gene for glutamate decarboxylase. Once researchers were able to identify how cells express specific genes, they began to look at how mutations in the genome affected the phenotype. But according to the National Human Genome Research Institute, the human body contains approximately 3 billion base pairs arranged into hundreds of thousands of genes. It's impractical to just wait for mutations to happen, so they went ahead and artificially created mutations in mice. For science. Using a technique called reverse genetics, neuroscientists were able to identify and manipulate certain gene sequences to create artificial mutations in mice and observe their effects. In fact, we've gotten so good at creating mutations that we've developed techniques like gene knockout and gene splicing to remove parts of a genetic sequence that code for specific proteins associated with diseases. One of the most popular findings in neurogenetics comes from a species of jellyfish that glows green when exposed to ultraviolet light. They're able to do this because of a gene that codes for a protein called green fluorescent protein. What's more interesting is that gene splicing allowed us to remove the sequence that codes for this fluorescent protein and genetically insert it into the DNA of other species. When a gene that doesn't naturally exist in a species is expressed, we say that it is ectopically expressed. So why does any of this matter? Because it's awesome mostly, but we've also used these findings to target dopamine neurons in the ventral tegmental area in mice, allowing us to separate them from the cluster of neighboring neurons, which is incredibly useful since neurons have similar features across the board. Neurogenetics is an incredible field of study, and we're at the cutting edge of neuroresearch, so we expect to see some profound advances in medicine and related fields.